The Cell Cycle by Morel, Harry, Rafael, and Bennet. The cell cycle, of course, begins with interphase. The G1 phase starts where the cell carries out metabolic activities to prepare for the S phase. At this point in the cell, all chromosomes are in a decondensed form in the nuclear envelope, which is contained within the cell membrane. The S phase is where DNA is replicated. Prophase is the next part of the cycle that also starts the process of mitosis. It is the longest phase of mitosis. In this step, the nuclear envelope disintegrates, and the decondensed chromosomes form condensed X-shaped chromosomes. The spindle apparatus forms as well. The metaphase is when the X chromosomes line up across the equator of the cell. Spindle fibers connect the centromeres of each chromosome. The fibers stretch from the poles to attach to the centromeres. Anaphase is when the centromeres are split apart and the formed spindle fibers shorten and thicken, pulling one chromatid on both sides of the cell. The cell begins to divide into two identical halves. Telophase is when the cleavage furrow forms, which is a thin connecting point between the cells. The nuclear envelope forms on both sides to contain the chromosomes. Finally, cytokinesis is when the cleavage furrow pinches to create two separate identical daughter cells. The chromosomes in the nuclear envelope decondense, and this shows asexual reproduction.